introduce and acknowledge Agnieszka Stadnik right there, and Christina Piedad, who really are at the forefront of our work with you. So some of the questions I will field their way, and as you know, many of you do not really have to wait till there is a webinar. If really there is something on your mind, and many of you I have seen already, please feel free to contact us. I know you, and will be able to answer your questions. So I know there are a lot of questions, so I'm not going to do a big presentation. I'm going to just give you some research updates, then also ask Agnieszka and Christina to give you just a little bit of their perspective of the sense that we have about communicating with you in these strange times, and how we're handling things. And then we'll leave the rest for questions and answers, which I know some of the questions are redundant, and I think Connie is going to do a very good job, as she always does, at fielding these things to us. So first of all, some research updates. I would be amiss if I don't tell you about what's going on with the atorvastatin trial. So the atorvastatin trial is doing extremely well. It would have been doing better and would have been probably nearly completed in terms of patient enrollments. Of course, the follow-up takes 2 years from the time you enroll, but we obviously had a slowdown during the COVID period. However, really the only shutdown was last spring, a year ago. So we have been enrolling patients also, although at a slightly more modest rate. But we have persisted with the enrollment, and we are pleased to say that we have completed 2/3 of the projected enrollments, and more than half the patients have already completed year 1 follow-up. More than half of the patients we want to enroll altogether. So we've enrolled so far 51 of the planned 80 patients, and 40 have completed year 1 follow-up. Many of the year 1 follow-ups are happening now, anytime, and I'll tell you a little bit about that. And we also have, I believe, 14 that have completed. Is it 14 that have completed year 2? You sent me those numbers.
11.
11 have completed year 2, which is great. We're starting to see more and more year 2, which means they got off the drug, and we await the analysis of their results. So just so you know, we had wanted to have 50 cases with full data sets, meaning people that complete the trial, don't drop off, make it for their follow-ups, and have a full set of imaging done at 0, 1 year, and 2 years. So if we have 50 of those, we wrap up the trial. So the problem is that in reality, in trials, some people stop taking the drug, some people drop off the radar, some people are hesitant about their follow-ups. So we end up having some drop-offs, and that's why we had built into the plan enrolling 80 instead of 50. So we have 50 with complete results. As it turns out, you are all doing better than we projected. So we, in fact, despite COVID, we reviewed those results today, despite COVID and despite all the hesitancy about travel and so forth, we, in fact, have more complete data sets than we would have anticipated. And another important thing is that the QSM and permeability, which you come to Chicago to have done the imaging, have been done at more than 97% success rate. In the past, we used to have 1 out of 6 of those images not go well, not be usable, maybe the head moved during the image or so. But we've been very good, and this is mostly because the team goes there. Many of you know, they meet you when you arrive. They're working with you during the MRI, and we call that the chaperoned MRI. So it's like a chaperone date. So we're chaperoning you, and we get you through it. But the result of that is that we don't have to have wasted studies. And I'm very encouraged that this summer we will do our interim analysis, and we may well end up deciding that we have enough data to open up and do the analysis. So we will see how that goes. The worst comes to worst, we need to enroll another 29 patients, but I really do not think we have to get there, because we have many good data in hand. Now, you need not to give up. And I have to be the preacher now. You cannot give up. You've been in the trial for 1.5 or 2 years. Now is not the time to say, well, I'm shy about the trip, you know. And you know, I will be calling you, and there are several of you I've called, and we've explained why it's important to complete the task, and many of you have come, you know, New York, North Carolina, Florida, Alabama. You come the night before, you stay 1 hotel night, and then you leave, and we give you a good stipend for the trip, and hopefully it makes it all worthwhile to know that we were able to look at you and make sure that we collected the needed data. It's not just about data. This feeds into healthcare during COVID crisis, and we have maybe a case a month that delayed their follow-up, and then when they show up, something had happened that they had blamed on maybe COVID, but they didn't have COVID. The guy who had dizziness, he blamed it on having been on a boat a month earlier. Well, it wasn't a boat; it was a big brainstem bleed, and so forth. So that we have had many cases like this, and therefore, please take care of yourselves. You have a disease. You do not have corona yet, most of you. So we need to treat you for the disease you have, even as we all take care for you not to get the disease that you don't have. So on the atorvastatin trial, we are doing well. Many of you have started to go back to Mayo Clinic, as well as us. So the trial readiness is picking up again. UC San Francisco has been essentially shut down. I think they're going to restart soon. Same with Hopkins. And same with New Mexico to some extent. So there has been some sluggishness in the enrollment in TR at those sites. However, for some reason, we more than made up for it between Mayo Clinic and Chicago. So our enrollments are very robust, and I think we're making up for the other sites. And we have a very good idea now, if we see 10 patients with cavernoma, how many of them would be eligible for a trial. And that's so important, because as we're proposing trials, people ask you, well, how many cavernomas do you see? And people say, well, I see 40 of them or 30 of them. But how many, after you go through the sieve, are ones that would be eligible for a trial? And that's what the Trial Readiness Initiative is all about. And that is going extremely well. The third and last project I want to tell you about from our site is the biomarker project, which many of you gave blood tests as far back as a few years ago. And based on that, we determined that we can look at certain molecules in the blood and predict if a person is going to bleed or not. And this was confirmed on repeated specimens and repeated groups of patients in Chicago. Now we have funding from the NIH to verify this, optimize it, and also check it whether it's valid at the Mayo Clinic and San Francisco. And we also have a parallel project in Rio de Janeiro, Brazil, so that as a result of that, we will know that that blood test is not some kind of fluke that only works in Chicago, but it is valid across places, and therefore may be able to be developed into a clinical blood test that you go when you have your MRI. You also get the blood test that tells you how is your lesion doing. Is it stable, or do you have maybe some lesions that are going possibly to be problematic with bleeding? All of us would want to know that, and all of you want to know that. So I think this is going to be a great project, and that project is enrolling also ahead of its rate, and we have started to have some very, very exciting preliminary confirmations there. So I think these are the updates on the human subject research from our end. I want to give a minute or two to Agnieszka and a minute or two to Christina if they want to add anything from their end or their perspective about follow-up with patients, about accessing patients, how are we open, how do we accommodate people, etc. Agnieszka, want to go first?
Sure, thank you. First, I want to thank everyone who has participated, for following up, and for sticking with us. We did create an app this year. As of, I think, January, we opened up officially. So we have an app now, which makes follow-up a lot easier. So there's a lot less shipment and check-ins. You can do it all through an app, and it'll remind you to take your pill every day, which we're very excited about. I think it's a great way for you guys to keep in touch with us, on top of obviously the study cell phone and emails and all that, which we always respond to. I do see a question about travel to the site being required. So for both trial readiness and the atorvastatin study, you need 3 visits. So a baseline, a year 1, year 2. You have to make those for the MRI that we can only do at Chicago. But other than that, all the check-ins are now virtual through the app or over the phone. So there's not a lot of travel for that. But yeah, not much to say besides what you already said, but just thank you to everyone who's helping us get this done, because it's such an important study and we're getting so much good information from this, and it's not doable without everyone participating and staying with it for the 2 years. So really just thank you to everyone. And if there's anything, as always, just reach out to us, and happy to answer questions or keep in touch or anything. So yeah, thank you.
Thank you, Agnieszka. Christina?
Yeah, thanks. I'll just reiterate what you both have said. Thank you first and foremost for your interest and participation in the studies. It's been a little bit challenging, but I think we've all risen to that challenge in a very good way. I feel very engaged still, and everyone has been in close contact, and we've been really trying hard to keep up with the follow-ups. If there is still any concern about travel, just talk to us, and we can figure something out. We still want the data, even if you can't come in a certain time frame. Just communicate that with us, and we can make whatever accommodations we can in this COVID time. And that's basically it. Aside from that, the app, yes, it has been very, very helpful. And we do see that people are using it every day and using it how we anticipated. So it's very helpful for us. At the same rate, the study phone is still active. So if there are any issues, please text or contact me. That phone is connected to me all the time and to any of the team members. So if there's anything that you need, just let us know.
Thank you. So yes, we are open and we're flipping burgers, so no problem. Now, let me tell you that this basically is going to get us very close to answer a question as to whether statins are decreasing the bleeding in the lesions or could potentially be dangerous and increasing the bleeding. I think it is an extremely important question that everybody wants to ask, not just to use statin, if we need to, but also for people who may need statin when they're older, etc. So it is very important for us to know what do statins do. We hope that they will be helpful. We do not know. We had a safety review, our second safety review of the trial, and it shows no red flags that would go against the drug. So it's either that there is very little effect to tell anything yet, or the effect is favorable. So we've been told to continue to enroll patients, so there have not been any significant safety concerns. The adverse events have been extremely low. So it's basically people, initially they have an idea whether they want to be on a drug or not, and they're nervous. But once they get on it, the people who actually develop symptoms from the drugs have been nearly zero. So it's been mostly, we worry about symptoms, we don't want to take the drug. That I can't argue with. But once you're on the drug, we haven't seen any significant problems. There haven't been liver problems, muscle problems, etc. So most people have carried on, have been good champs about it. Now, as we update, you all know that there have been a couple of papers out about the effect of propranolol in mice. Actually, our paper came out about a month ago. It was a very, very ambitious project that started with zebrafish, working with the San Diego group and looking at propranolol in the fish. And we were able to tell how propranolol might work, if it's going to work, and that it works actually through the slowing of the heart and lowering the blood pressure mechanism, which is different from the mechanism in which it works in the infantile hemangiomas, where people have big hemangiomas on their neck. In those cases, there is a different signaling that seems to be at play. Propranolol looks like it has an effect that is the same or weaker than statin. It does seem to decrease lesion growth, but did not affect bleeding. And this is the same result that just came out from the Italy group. Their model did not include bleeding. We had two models with robust bleeding, so we were able to look at bleeding. And of course, the Italians did not have the prior studies with statin and Fasodil to compare in the same model, which we published. So we're able to tell head-to-head how these drugs do. So where we are left now is that propranolol should not be given up on, but we quite don't know how to use it and whether it really will affect bleeding, and or it's just an issue of lesion growth, and which lesion growth happens not very commonly without bleeding. So this is an important thing that will be studied. There is an ongoing trial that has finished enrolling more than 60 patients in Italy, and they are going to start looking at signal effects. Now, that trial is not going to have an endpoint like the atorvastatin trial. It's not powered with X number of patients to see an effect, yes or no. It is more of an exploratory trial to see if there is a signal. So if they see a signal, then that will be encouraging. If they do not, we have to go back to the drawing board and try to see at what dose does propranolol need to be used. Our mice studies required a huge dose that most of you would not tolerate. If you took it, you'd get dizzy when you stand up, blood pressure would go down. So that's not a good use of propranolol. And when you use propranolol at a very low dose, it doesn't have the beta effects, and therefore we do not know if it's going to work. So we are in the process right now between Dr. Marshak and my lab doing low-dose propranolol in the same mice to try to see if we see an effect of low-dose propranolol. And then maybe propranolol will be the next drug we test after statin. It is a repurposed drug. we can use the same platform as the trial readiness to test it. So that is the update on propranolol. Should you use propranolol or not? You know, it's a drug that's approved. People have used it. I think the only downside of asking your doctor to use it is a false sense of security. We have had many people take it and then get very disappointed that it didn't do anything, or many people bled despite being on propranolol. So you're taking basically an untested drug, and don't get a false sense of security that you're getting some panacea, because we don't know. We really don't know what it does and what it doesn't do. I don't currently prescribe propranolol because I do not really know if it's going to help. But if people are on it for blood pressure, for headache, we encourage them and we'll see if it works. We have a number of patients on propranolol in the trial readiness study, and we have not seen a signal that those patients are bleeding less than the others. But that's a bleeding trial, and we're seeing just as many or a little bit more bleeding on propranolol. So the answer is not out on that, but it's exciting that that's a common drug on the shelf. The final drug that I want to tell you about is the drug under the name Tempol, but has another name with the letter and numbers, and makes it sound very exciting because that's the oral form of Tempol. Tempol was initially a drug used on the skin for hair loss, but that drug has effects via the similar pathway as vitamin D, and it had some effects in mice that were shown almost 10 years ago. There have not been much more studies in mice, and we do not have any head-to-head comparison. But what we do have with that drug is a drug company that is absolutely committed to test this drug, and if it works, to develop it for cavernomas. So we don't have any other drug right now with a company that has millions of dollars ready to invest. Therefore, I think it is going to be very important for us to engage that trial. We're working with the company to try to make sure that that trial has the greatest chance of enrolling and succeeding. And we will have more to tell you about it, probably not very long, in the next few months. They had hoped few weeks, but nothing works like that, especially when nobody travels and half the sites don't even see patients still. So I think it will get there, and that will be another option in terms of being in a trial. But again, you're going to have placebo or the drug. It's a drug never used in man before, in humans before, in man or woman before. So we have to be very cautious with it. It's not like statin, where we know 50,000 people have taken it. But yet it seems that in the few patients that had it so far, it seems to be very benign and well tolerated. So I'm very full of excitement about that, and we'll tell you more about it. The last thing that I want to tell you about the research is that very soon we're doing the final ironing of the paper. There's going to be a huge paper in Nature that is going to show the mutation that drives the development of cavernomas, even when you have a solitary lesion in the vein. So before we knew the genetics of the familial disease, and we knew that the same mutations happen in the sporadic lesions, but we didn't know how it ties up to the vein, and whether the mutation in the vein drives the cavernoma or not. We now have very, very good evidence on that, and I'll tell you more about it after the paper comes out. But why is that important? Because that's another way to use a drug to target that particular mechanism. And there are some drugs out there that have very, very favorable effect that maybe could get us to the point of shrinking a cavernoma, not just preventing one from bleeding or growing. So very exciting progress on those fronts. I think I stayed at about 25 minutes, and let's go to questions.
Sure. So obviously the big question right now, well, actually, let me back up a little bit and we'll get to some of the questions that relate to what you've already spoken about. Someone is asking if you could perhaps explain the blood biomarkers a little bit.
Yes.
Are they just for people who have already had a bleed, or what is the purpose of blood biomarkers?
No, so the blood biomarkers, the beauty of them is that they apply to people who haven't bled to tell you that your lesion is stable and won't bleed next year. So what that blood is it combines, so far we have 4 molecules in the test, but we think ultimately it will be 5 or 6 molecules that bring it up to almost 100% precision. Right now, we're about 90% with 4 molecules combined. So it's like having 4 blood tests and we add the formula. That you test your sugar, your sodium, your cholesterol, you add them up. It's not those molecules, but we look at 4 molecules. How the hell does that do that? That's because when we are bleeding, there are signals that go from the lesion into the blood. And also when we are ready to bleed, when a lesion is ready to bleed, is ripe, there are specific genes activated and specific signals. We've been studying this for more than 6 or 7 years now, and it's real. And the beauty of that is that not only do you get your MRI, you may not even need an MRI. You just get the blood test and tells you your lesion is okay. It hasn't bled, hasn't done anything last year, and is not going to do anything next year. So if we're close to 100%, that's pretty damn good. That test, I know every one of you will want to use. So when we're talking to companies about developing the test, they ask me, how do you know the patients and the doctors will use it? I'll tell them, develop it and they will use it. I promise you. There is not one that doesn't want to know if their lesion has acted out or is going to act out. So that's what we're after to try to solve.
Okay. And just so that the Q&A doesn't get all backed up with, we will be going on to COVID in just a little bit, but we're going to ask the questions first that have to do with the other research. So,
and you know, the COVID questions, many of them overlap. It seems like we have 80 questions, but at the end, they'll be pretty,
Yeah. And so the question, still with the blood biomarkers, is this something that someone was asking, can they go to NIH and get this test? Where exactly can they do blood biomarker testing? It's not approved for clinical use, it's in research, right?
So currently the sites that are participating in this study as part of the NIH trial are Mayo Clinic with Dr. Fleming, UCSF Center of Excellence, and Chicago in the US. If you want to go to Brazil, you can go to Rio, you'll get it there. So we have those 4 sites because we want to be sure that it is applicable at multiple sites before it can be approved by the FDA and before a company will develop it. I am already working with the business school at the University of Chicago about companies adopting that test because we really need to move it forward to production. But it's one thing has to be done first. So if you are at one of these 3 centers of excellence, you can have the test. You will not have the results. You can have the test. We are in the process because we cannot tell you, I cannot tell you you're going to bleed or not when I'm not sure the test works. So we will, but you can participate and help develop the test.
Okay. And then the question is, for clinical trials, folks are asking, we have Australians in the watching tonight, we have Europeans watching tonight. What can they participate in?
Yeah, so far, unfortunately, there is no international trial going on. There is a trial in Italy, so you have to take the train and go to Lombardy or something. But other than that, it's the US trial. Those are the only 2 trials actively enrolling right now, Chicago or there. Now, we enroll international patients. We have people from Canada, people from Brazil, etc. So it's not forbidden. It has to be that. What are the rules currently, Agnieszka, about international patients? First, you have to come to Chicago for 3 visits 1 year apart. So a trip a year for 3 years.
Correct.
Initial 1 and 2. And
Yeah.
then what else?
That's really it. I mean,
English. I think just if you speak English, if we do not have it translated except into Spanish, correct?
correct. The other issue is sending the medication. So
Right.
we found a way to get it to Canada. Other countries might be more difficult.
Right. So basically, if you're coming from place like Australia or Brazil or whatever, if a family member in the US will send the medication to them, and then they can get it to you. So, and the app works. So you can enroll, but unfortunately, right now we are at a stage where the trials require traveling to the site. That includes Italy. So they cannot like do it remotely to Italy because everybody's using these biomarkers and they can't be done everywhere. So they want to know if the lesion is bleeding, whether you feel it's bleeding or not. Now the TEMPOL trial may be open to enrolling large enrolling sites overseas. But we're going to have to see if the sites have enough patients to be worth being brought on.
Okay, and then that leads to the next question about the TEMPOL trial and enrollment criteria. Will they be different? I know that they're still sort of being worked out right now, but
Yeah, it'll be slightly different. The latest rendition is that if you're eligible for the CASH trial, you can get into either the atorvastatin or the TEMPOL trial. If you are already on statin for cardiovascular indication, you can't be in our atorvastatin trial. Or if you're closer to a site that does TEMPOL, you just enroll into the TEMPOL trial. If you come to Chicago, we will offer you both. Now, it will be also open to people whose lesion is symptomatic, but not recently bled necessarily. So we're still trying to work this out because this sounds like you know it when you see it until you start combing through what that really means. Because many of you have symptoms for many, many years, and many of you have had symptoms of a past bleed or a past operation. And we don't want to downplay those symptoms, but that's not a symptomatic cavernous malformation. That's a cavernous malformation that hurt you in the past. So we're trying to see how is the best way to separate the new attributable symptoms from that. They wanted to include the seizures initially, but the FDA told them seizures are treated with seizure medications, that they didn't want to confuse the effect of TEMPOL on seizures. So as a result of that, seizures will be a qualifying event, meaning if you're symptomatic from a seizure, you can still enroll in it as a symptomatic cavernoma, but seizure is not the endpoint. So that's kind of what we're sorting out. Not final.
Can I also add that this is a phase 2 trial that's going to be starting? So phase 2 trials are typically much smaller. If the phase 2 trial is successful, then they should be going on to a phase 3 trial, which is much larger and broader, and probably the enrollment criteria will be also widened out to include more of our patients. Is that fair?
And easier to enroll at multiple sites.
Easier to enroll at multiple sites.
Because you know, you may not need to use biomarkers in the phase 3. So we'll have to see.
And so someone had asked if they'd had brain radiation, would that make them ineligible for the TEMPOL Rec 994 trial? And for the phase 2, I believe it would, but who knows with the phase 3, we don't know yet what those criteria will turn out to be.
Yeah, and the radiation is a problem because some of the lesions are real cavernous malformations. Other lesions are radiation bleeds or radiation hemorrhagic angiopathies, and they don't have the exact same mechanisms. So it's every trial at phase 2 doesn't want to dilute it with cases with radiation that may give false results.
Okay. Next question is a very broad one. How is, and very important, how is symptomatic defined?
Yeah, That's a big hang on. It may end up at the end of the day going back to bleeding or growth or focal symptoms with growth, but we already in cash accept focal symptoms with growth. So the problem is if you have focal symptoms but no growth, that may be allowed, but they have to be new focal symptoms attributable to a lesion. So we can't have somebody come in and say, I feel tired and I'm depressed and it's because of my cavernoma. We have to be able to see how is the cavernoma linked to the symptoms, or the FDA is not going to buy it.
Okay. And then back to the biomarker trial, just real quick, how much longer do you need to test the blood biomarkers? How long will that trial go on?
So we have just received the 5-year grant to develop it completely. That doesn't mean that we do not start working with a production company or with the FDA beforehand. So I am very, very encouraged that within 5 years, a form of this test could be ordered by any doctor. So that's going to be faster than a drug.
Okay. And then I think we have a few more questions, but just one more that's actually on medicine. Do you know what dose level is being used in the Italian propranolol trial?
Yeah, so they're starting low and raising it, and most of the people are getting between 20 and 40 milligrams, which is what most people tolerate for migraine and for other things. If you take propranolol for blood pressure, you usually take 80. So if we have to drive it to the level where it affects blood pressure, it's not going to be very well tolerated, and it's not being tested at those high doses in Italy.
Okay. All right. And now, different topic. Folks have been exposed now to Dr. Fleming's recent paper on CCM and pregnancy.
Yeah.
Can you make any comments about hemorrhage as a risk factor in pregnancy, and if you know anything about her paper?
Yes. I know a lot about her paper. So basically, what it showed is that in general, pregnancy does not pose an added risk, which we've been telling everybody that. However, there are some bleeds associated with pregnancy. So we can't exclude that the bleed will happen during a year of a woman's life when she is pregnant. And then there is the effect of estrogen and progesterone, which is in another paper that came out this past year, that links the use of estrogen and progesterone. We have long suspected this, but we haven't had a very good way of proving it other than anecdote. And now there is evidence in a paper that actually people who are using exogenous estrogen in particular, there were not enough patients with progesterone, exogenous estrogen have more acute hemorrhage. Recurrent hemorrhages are still questionable, but acute presentation with hemorrhage seem to be more associated with that in women with or without estrogen. We have many, many cases. Because I see so many patients, I can tell you there is not 2 weeks, 3 weeks that go by that we don't have a bleed where the only factor we know is the new starting of estrogen or progesterone medication or in vitro fertilization drugs. So I'm convinced that they do have some activity. How much, and can you get away without a bleed? Yeah, you can get away without a bleed, but it does seem to be associated with it. I wish we knew more about the mechanisms of that.
Okay, thank you. All right, let's talk about COVID. First off, my personal question is, what do we know now from the COVID registry, from folks who have CCM and tested positive for COVID?
Right, so let me... Right. So the knowledge depends on what the registry has in it. So let me tell you what we have. So this registry depends on people reporting that they have had COVID and they have a CCM. And the COVID has to be PCR or other test proven. So if you had the rapid test and antibody test or PCR, we accept that, but we're not going to accept, I think I had COVID, but I can't prove it, you know. So it may be limited because a lot of people with benign, asymptomatic or minimally symptomatic COVID don't get tested sometimes. And there is that error. Another error is the reporting error. I don't know if I have all of you in the registry. Okay, so that is, and more importantly, do the people who are not reporting, are they more likely to have more severe disease and they're so sick they're not reporting? So that is concerning. I'm not worried about the mild because the registry is showing no problem. Okay, so we have about 40 of you so far who have cavernoma and COVID. And there is no hint of that the COVID is more severe or that the CCM disease is more severe. We do have a couple of bleeds. One happened before the patient knew they had the cavernoma, so that happens anyway. And there is one other bleed that happened several months after COVID. So there is not a lot of my lesion blew out because I had COVID, but that's the limitation of the registry. And I'm going to actually, before we report these results, I'm going to be asking Connie perhaps to do another anonymous survey without any names or without even joining the registry to know if we're missing anybody who was severely affected or died from COVID and the family didn't report, who we knew have cavernoma. Even if I know that there are such patients, then we know that our registry doesn't say anything about them. But if Connie asks everybody and you guys, none of you know anybody, that gives us a greater comfort level that we're capturing really the illness. Of the 40 or so cases, about 4 to 6 had fairly severe COVID. And therefore, that's about representative of how COVID plays out when you have it.
Yeah. And I think just thinking about our patients who communicate in the forum and knowing patients in the New Mexico area where there is a greater density of patients, I think the severity that we see, and particularly in New Mexico, is age-related, just like it is in the regular population. The people who have been getting sick or perhaps even passing away from COVID are the elderly.
Yeah, so if we know of anybody passing away, we need to kind of get some hint of that, because otherwise that registry is meaningless, you know, if it's sieving out only mild.
I will, but I think we'll find most of those people in New Mexico, so they may be a little bit harder to get to, but I will.
And of course, there you're going to have a population that has COVID and they have CCM, and some will die from COVID, so we won't know that their CCM had any bigger problem. Another way we looked at it, you know, because our site sees so many cavernoma patients, and also we had at one point many COVID patients, we would have detected if somebody didn't know they had their cavernoma and that they're having significant issues. There is, again, that one case we know of that bled, and then when we removed the hematoma, there was a cavernoma in it, but the patient didn't know they had it.
Okay. All right, let's talk about vaccination. That's the big question that everyone has right now. The general question is, should people with CCM get vaccinated? Is it safe for them to get vaccinated?
Yeah, so we put a statement about this, anticipating that all of you will be asking that question. We even put that statement when we knew the trial results, and the FDA was just in the process of reviewing those trial results, even before there was vaccine available. So the argument started before there was anything to argue about. But you know, I am not a COVID expert and I'm not a vaccine expert, but I am a trialist. And I have studied the trial data very, very seriously with an eye toward the cavernous malformation. I have other patients with aneurysms, etc. So there is no question that the vaccine protects you from severe COVID, from getting hospitalized from COVID, or from dying from COVID. There is very good evidence that the vaccine also protects you from catching COVID. The reason we know that, although that wasn't studied in the Pfizer trial, it was studied in the Moderna trial that has the same mRNA type vaccine. And there was a halving of the likelihood of even carrying the virus. So not only do you protect yourself, but you protect your loved ones if you're vaccinated, as best as we can tell. Do you pay a price as a result of vaccination that may not be worthwhile? That's what trials are all about. And trials have looked at this very, very carefully. And number of deaths, number of illnesses, number of hospitalizations, all of that is 5-fold less in the vaccinated group than in the non-vaccinated group. Every death on the vaccine trial was analyzed, and there were deaths of people who had the vaccine from a heart attack, but there also were deaths from a heart attack in the people getting placebo. So you're going to have situation where association will say, I had the vaccine, I had a heart attack, look what happened to me. But when 40,000 people are studied, there is not that increase. The one increase is allergic reaction if you are susceptible to severe allergies, meaning you carry a pen with you all the time and you've had anaphylactic reactions. Nobody has died from that, believe it or not, but it has happened and they've had to use their pen. And that's why now with all of these vaccines, you're asked to sit for 15 minutes after you have it. And if you have a history of allergies, you're asked to sit for 30 minutes. And with those practices, nobody has that problem. So that's what we know. We know that, yes, you should get vaccinated and there is no reason not to.
Do we know anybody yet with CCM who has been vaccinated and had significant adverse effects? For
No.
example, a hemorrhage?
No, we know many people with CCM that have been vaccinated. We have written letters for people to get in line because they ask, and I know this will be a question, is CCM a condition, a medical condition that should put me priority in the vaccine? We'll comment on that in a second, but the fact of the matter is I am unaware. I would love to be aware if anybody has had a genuine proven CCM related problem or other problem with the vaccine. We'd want to know. We do not have it in the registry. I don't think I have enough scientific evidence to create a registry for that, but if there is 1 or 2 cases, I would like as a doctor to get involved to help you with that. And then as a result, we'll start a registry or a study. But so far, thousands of people are getting it. The whole University of Chicago Hospital got it. Nobody dropped dead from it.
Yeah, I can tell you, my daughter who has a CCM3 mutation got her second shot yesterday. This morning, she woke up with feeling achy and she had a fever of about 100. By midday, she was good. So that's,
yeah, the second dose,
that was
the second dose for those 2-dose vaccines, the second dose gives you a little bit more of a feeling of, I think Christina had quite a thing with it. She had to take Motrin. I didn't. I had no issues. Agnieszka, did you have any issues? Second dose a little bit?
Really tired. That was it.
Yeah, just a little bit tired. The second one seemed to hurt a little bit more than the first one, but you know, I'd rather have that than having COVID.
Yes, absolutely. Okay, and so then the question again that you started alluding to is, should people with CCM be prioritized for vaccination?
Yeah, so we talked about this at the advisory board, and I think all of us feel that if you had seizures, a prior stroke, prior brain surgery with a deficit, a case can be made that you have a condition that, you know, if you had COVID and have seizures, maybe COVID would make the seizures worse. So I am willing as a doctor to say that this is a medical condition that should put you ahead in the line.
Okay,
And we've been helping our patients with that.
okay.
But if
So
you're
that's-
asymptomatic and you have a single lesion in your head and you're asymptomatic, I don't think we can make the case that you have a medical condition that requires you to
okay.
move ahead.
So then the next question is, does it matter which vaccine you're getting? One of the mRNA vaccines versus AstraZeneca versus the J&J one that's coming down the line, are there extra risks with any of them, or are there any advantages?
Yeah, so there is some comparative effectiveness data, although all those trials are not done exactly the same. It's similar to like what we do with the mice, one done in Italy, they do it a little bit different and so forth. But basically, the mRNA vaccines, Moderna and Pfizer, that first hit the market, have the most effectiveness against serious illness or death from COVID, effect near 95%. So it basically shuts down severe illness. And it not only worked in the trials, but it has worked now in the nursing homes, where the most vulnerable people were the ones who received the vaccine, and it basically shuts down COVID in the nursing homes. So it is a very, very compelling effect. The Johnson & Johnson, that is the third one to be reviewed by the FDA in the US. It hasn't yet been approved, but it's on a track to likely being approved, is a single dose. So that's kind of advantageous. Also, it's a vaccine that does not require -80 or -30 for storage, so it's going to be very good to use. But the data is showing 75% effect. Now, epidemiologists love that. They think in the population that's great. But if I had a choice between two vaccines, I'd get the one that has 90-95. But my recommendation is going to be, we don't have the luxury. In this country, there are more people that need the vaccine than there are vaccines. So we can't fight over which one is better. My recommendation is get whatever vaccine you get. The first one that would have arrived to University of Chicago, I would have taken. Now, the Chinese and the Russian ones are a little bit problematic because the data is not published in a way that can be really analyzed. We have to trust the Chinese government and the Russian government that they work to the same extent. And they are produced using the classical vaccine methodology, meaning they have pieces of deactivated virus. That virus will not cause an infection, but they are not as precise. They're not precision medicine driven like the mRNA vaccines. So that's probably why a lot of the effects are in the 70%, 80% when you go into this classical vaccine. I'm going to go into something that you were probably going to ask, but has to do with, is this mRNA some genetic fluke that we should trust? Or are we making our body produce proteins? Are we going to turn into apes or, flies or whatever? This is the most precise way to target the virus. Even if you took pieces of virus, I wouldn't even know what that really would do in terms of some pieces being potentially toxic, etc. However, when you have that very pure form of synthesizing the one protein that actually triggers the disease and fighting that protein. The protein alone will not trigger the disease. It's when it's with the virus that it triggers the disease. However, if you have the protein there, you create antibodies to it. When a virus comes with that protein, they attack the virus. So it's a very, very cool way of not introducing a virus into your body, but yet fooling your body to know that you may have a virus, therefore it produces the immunity. Now, is it a new thing, and therefore maybe next year I will grow horns because of it? Every vaccine, you do not know the effects 5-10 years later until you do post-approval surveys. However, with these mRNA vaccines, we are lucky because they were developed for a disease called SARS. You may remember it. It's also a coronavirus that caused respiratory disease, and also it caused Middle East coronavirus, Middle East respiratory syndrome. And that's when they developed the mRNA vaccines. So we actually know that they had it tested for safety. It's just they never got into widespread use because those diseases died off. So they were prepared to create these mRNA vaccines on a wide scale, but luckily those first two corona attacks 10 years ago and 15 years ago never became a pandemic. So we never had to deploy the vaccines on a wide scale, but we have evidence that they are safe from before. And now we have the trials with the new vaccine. Nobody will tell you if the vaccine is gonna do something 5-10 years from now, but COVID will at a much higher rate. We know that for a fact.
And then there's like a half a dozen people who have said, so how do I get something that says I can get on the priority list? And we have someone asking from.
Yeah, I think you need to talk to your doctor. Whoever your doctor, they will write you a note. You know, you have a condition, you had a stroke, you have epilepsy. All what they have to do, they don't have to lie. They don't have to say you need to get the vaccine. All what they have to say is that you have a medical condition.
Yeah.
Say what it is. And that's enough for most places to move you up in the line and for you to self-declare as having a condition.
And CDC lists cerebrovascular conditions. So
Yes.
that's what we
Yeah,
have.
I mean, this is, we are not cheating the system. You are not standing ahead of anyone else. It is legitimate. Now, if you have something that's asymptomatic, I think it's less compelling. If you can get vaccinated, more power to you, but I won't write you a letter if you're asymptomatic, because I really have evidence that COVID is not going to do anything to you. So you are like anyone else.
Okay.
But if you had symptoms and you have COVID, maybe you get more seizures, maybe your respiratory problem is worse because you've had prior stroke. So I think that's compelling.
Okay. A couple more quick questions. Someone has already had COVID. Should they go ahead and still get the vaccine?
Yes, so all the trials have included people who have had COVID before. Some of them required them to wait 3 months before they take the trial. Some enrolled them right away. And that subgroup was several hundred patients, and they benefited to the same extent. So although you've had COVID, you are less likely to catch COVID again. With the vaccine, you are 5 times lesser likely to catch COVID again. So yes, the vaccine will help you even if you had COVID before.
Okay. And last COVID question, but then I need to drop back to an IVF question. COVID question is it true that you should not use NSAIDs before and after the COVID vaccination?
You know, there's been talk about that because it might inhibit the immune response and was not tested in the trial that you take it. But also we do not know you shouldn't. I think this is kind of a theorizing that people bring up. In fact, I can tell you that I took Motrin right after mine because my arm was hurting. So I don't know what's beeping here.
Dr. What is your building burning down?
I think my wife is cooking something. She set off the alarm.
Oh, no. All right. Well, the last question, because we're almost at the top of the hour, is there are 2 people who have asked again about IVF. Is that something that women with CCM should be avoiding? Does it matter whether you have CCM1, sporadic, CCM3?
We have seen a new bleed with all types of CCM after IVF. That doesn't mean that that same patient wouldn't have bled if they didn't have it. So I cannot tell you it was absolutely caused, but the association is so compelling that I think you have to be very closely watched for for your cavernoma. So our practice is that if you want IVF, you need to be aware that it might cause a bleed in your CCM. You should have your CCM checked for stability before you have IVF. So don't have an IVF if your CCM just bled, or if you don't know if it just bled, because then you are really probably setting it up for something else.
Okay. Thank you. Thank you. I think
Thank you so much, everybody, and hang in there. The end is in sight with the COVID, and the end will be coming with cavernomas. We're working very hard.
okay. And I need to let everyone know that we have support group tonight in the chat. I'm going to leave that open for a little bit longer. Is the link to the support group. It'll start immediately, like now. So if you want to click that link and just move over to that Zoom, please feel free to.
And I want to again thank Agnieszka and Christina for joining us after their hours. They're always there for the CCM community, and thank you, everyone. Thank you, Connie.
Thank you. Okay.
Thank
Good
you.
night, everyone.
Bye.
Bye-bye.